Welcome students. Now we are moving to nitrogen cycle and nitrogen metabolism. First, nitrogen cycle. In nitrogen cycle, there is 5 steps. First one, nitrogen fixation. Second one, nitrification. Third one, nitrate assimilation. Fourth one, ammonification. 5, denitrification. So first step is nitrogen fixation. So we already discussed about nitrogen fixation. The atmospheric nitrogen is converted into ammonia is called nitrogen fixation. So nitrogen fixation requires some requirements like first one, nitrogenase enzyme complex. Nitrogenase enzyme complex. So nitrogenase enzyme complex is required for nitrogen fixation, and also minerals like molybdenum, iron, sulfur. Molybdenum, iron and sulfur is required for nitrogen fixation, and also it requires anaerobic condition. Anaerobic condition and it requires energy, requires electron, and it requires an H plus donor. It requires an H plus donor. So nitrogenase enzyme is active. Nitrogenase enzyme is active only in the presence of anaerobic condition. So the anaerobic condition is created in leguminous plants by the presence of a pigment called lead hemoglobin. So the presence of lead hemoglobin in the leguminous plants will make the anaerobic condition. How this pigment lead hemoglobin make an anaerobic condition means this lead hemoglobin may act as a oxygen scavenger. They act as a oxygen scavenger. They act as a oxygen scavenger. They remove the oxygen from the nodules. So the nitrogenous enzyme is active only in anaerobic condition. The anaerobic condition is created in the nodules by the presence of a pigment called lead hemoglobin. This lead hemoglobin may removes the oxygen from the nodules to create an anaerobic condition. So the lead hemoglobin may act as oxygen scavengers in the nodules. So the nitrogen fixing bacteria in root nodules. The nitrogen fixing bacteria in root nodules is simply rhizobium. So the rhizobium appear as pink in color due to the presence of this lead hemoglobin. So these are about nitrogen fixation. We already detailedly studied about nitrogen fixation. These are the requirements for nitrogen fixation. First one, nitrogenase enzyme complex. Then the minerals like molybdenum, iron and sulfur. And anaerobic condition, ATP, that is energy is required, then electron and X plus donor. Then the nitrogenous enzyme is active in the presence of anaerobic condition. So the anaerobic condition is given to the root nodules by the presence of the pigment called lead hemoglobin, and the lead hemoglobin acts as an oxygen scavenger, and they remove oxygen scavenger from the root nodules. The second stage of nitrogen cycle is nitrification. The second stage of nitrogen cycle is nitrification. It is the conversion of ammonia, that is the nitrogen fixed ammonia is converted into nitrite form. NO2 minus is nitrite and NO3 minus is nitrate. Should not be confused. This is the plant absorbable form. NO3 minus is the plant absorbable form. So from N2 we have to convert it into NH3, that is ammonia. From NH3 we have to convert it into nitrate. From nitrate we have to convert into nitrate. So this nitrate may be utilized by plants for the metabolic process. So the conversion of ammonia into nitrite and the conversion of nitrite into nitrate is called nitrification. Simply the formation of NO3 minus and NO2 minus is the nitrification. So the formation of nitrite is 2NH3 plus 3O2. Before that, there are some bacteria which helps in this formation. So nitrite formation and nitrate formation. So in nitrite formation, the bacteria Nitrosomonas is converted that ammonia into nitrite, whereas in nitrate formation, the Nitrobacter bacteria helps in the formation of nitrate. So it is Nitrosomonas here it is Nitrobacter. So these bacteria plays an important role in the formation of nitrate as well as nitrate. So 2NH3 plus 3O2. In the presence of Nitrosomonas give 2NO2 minus plus 2H plus 2 molecules of water. This 2NO2 is again converted to 2NO2 minus plus O2. In the presence of Nitrobacter give 2NO3. So this is simply nitrification. Now we are moving to the third step, that is nitrate assimilation. Nitrate assimilation. So the process by which nitrate is reduced to ammonia, that is the process of reduction of nitrate into ammonia is called nitrate assimilation. So the the enzymes like nitrate reductase are responsible for the conversion. So we can detail a look. First one, the NO3 minus converted to NO2 minus in the presence of nitrate reductase. So, here molybdenum, it is a micronutrient. It acts as a cofactor. So the nitrate is converted to nitrite in the presence of nitrate reductase with the help of the cofactor molybdenum. Now this nitrite is converted to ammonia with the help of nitrite reductase. Here, iron and copper act as a cofactor. So, this is simply nitrate assimilation. Then, the fourth step is ammonification. The fourth step is, ammonification. Ammonification is simply the decomposition of organic nitrogen from dead plants and animals into ammonia. So, the organic nitrogen, it can be from plants and animals. Dead plants and animals. The organic nitrogen is converted into ammonia is called as ammonification. For this ammonification, some organisms may involve. Example, Bacillus ramosus and Bacillus vulgaris. These are the two organisms which takes part in the process of ammonification. So, the organic nitrogen is converted to ammonia. This is called ammonification. Then the last step is denitrification. The last step is denitrification. Denitrification is simply when the nitrates in the soil are converted back into atmospheric nitrogen. So, nitrate in the soil, NO3 is converted to atmospheric nitrogen. It is called denitrification. The process involves some bacteria called denitrifying bacteria. Denitrifying bacteria, those are Pseudomonas, Thiobacillus and Bacillus subtilis. Pseudomonas, Thiobacillus, Bacillus, so these are the denitrifying bacteria which helps in the process denitrification, which is the conversion of nitrates in the soil into atmospheric nitrogen. So these are the 5 major steps involved in the nitrogen cycle, and with the help of these steps the atmospheric nitrogen is fixed into nitrate and it is absorbed by the plants. Now we can look nitrogen metabolism. Nitrogen metabolism or otherwise fate of ammonia. Fate of ammonia is of three. First one, reductive amination. Second one, transamination. Third one, catalytic amination. Reductive, actually the ammonia is converted into many amino acids by these reductive amination, transamination as well as catalytic amination. First one, reductive amination. What is the reductive amination? Reductive amination is the glutamic or glutamic acid is formed by the reaction of ammonia with alpha ketoglutamic acid. So, alpha ketoglutamic acid plus ammonia, the presence of glutamate dehydrogenase one molecule of energy is used. It forms glutamate plus water. So, reductive amination means the ammonia combined with the alpha ketoglutamic acid in the presence of glutamate dehydrogenase gives glutamate. Glutamate is an amino acid. It is an amino acid. So, this is reductive amination. So, the ammonia is converted to some amino acid. Second one is transamination. Transamination. In transamination, the amino group from glutamic acid is transferred to keto group of keto acid. So, the amino group from glutamic acid is transferred to keto group of keto acid. Almost all the amino acids are formed from glutamic. So, glutamic acid is the main amino acid from which other amino acids are synthesized by this process, transamination. So, transamination requires one enzyme and a coenzyme called transaminase and pyridoxal phosphate. Transamination requires transaminase is an enzyme and a coenzyme called pyridoxal phosphate. So, the amino group from the glutamic acid is transferred to the keto group of keto acid. So, amino group is transferred to keto group. It is from the amino acid to the keto acid. So, this is simply transamination. Then the last one is catalytic amination. Last one is catalytic amination. also called as go-gate pathway. Go-gate pathway. GS go-gate pathway. In this amination, what happened is the glutamate or the glutamic acid combines with ammonia to form the glutamine. That is glutamate combined with ammonia in the presence of glutamine synthetase using 1 ATP molecules, it will give glutamine. So this is simply the first, that is GS pathway. Why it is called as GS pathway? That is here the glutamine synthetase is using, that's why it is called GS pathway. Next pathway is go-gate pathway. Go-gate pathway. The enzyme here used is go-gate. It is glutamate 2-oxoglutarate aminotransferase. Glutamate 2-oxoglutarate aminotransferase. Enzyme which is responsible for this reaction. That's why it is called as go-gate. The reaction is the glutamine which is synthesized by GS pathway is combined with the alpha-ketoglutarate acid in the presence of go-gate enzyme to form 2-glutamate. So, glutamine plus alpha-ketoglutarate acid, the presence of go-gate enzyme give 2-glutamate. So, this is the go-gate pathway. So, these are about the nitrogen metabolism. Nitrogen metabolism, that is the fate of ammonia is reductive amination, transamination and catalytic amination. Now, we are going to discuss special mode of nutrition. The last topic of our chapter, special mode of nutrition. Special mode of nutrition. We know that nutrition is the process of uptake and utilization of nutrients by living organisms. There are two types of organisms or nutrients, that is autotrophic nutrition and heterotrophic nutrition. Autotrophic nutrition and heterotrophic nutrition. So, what is autotrophic? What is heterotrophic? Autotrophic is simply the process where an organism prepares its own food from a simple inorganic materials like water, minerals and carbon dioxide in the presence of sunlight. So autotrophic means auto means self. So the organism prepare their own food by using some simple minerals like water, minerals, carbon dioxide in the presence of sunlight. Then what is heterotrophic means? The organism which depend upon other organism for food and survival. So auto means self, they can prepare their own food, whereas heterotrophic means they depend other organism for food. In autotrophic, it is again divided into two, photosynthetic and chemosynthetic. Photosynthetic and chemosynthetic. Photosynthetic means the organism prepare their own food in the presence of light, whereas chemosynthetic means the organism prepare their own food with the help of oxidation of substance, of chemical substance. So that's all about autotrophic nutrition. Autotrophic nutrition means simply the organism prepare their own food by using water, minerals and carbon dioxide in the presence of sunlight or with the use of chemicals. The autotrophic nutrition are of two types, photosynthetic nutrition and chemosynthetic nutrition. Photosynthetic nutrition is organism synthesize organic materials using light energy, whereas chemosynthetic means organisms synthesize organic materials using energy derived from the oxidation of inorganic chemicals. Now, look into heterotrophic nutrition. Heterotrophic nutrition include saprophytic nutrition, parasitic nutrition, symbiotic nutrition and insectivorous nutrition. So, heterotrophic include parasitic nutrition, saprophytic nutrition, insectivorous nutrition and symbiotic nutrition. So these are the four types of heterotrophic nutrition. Now we can detail study what are the saprophytic mode of nutrition. Saprophytic mode of nutrition. Saprophytes derive nutrient from dead and decaying matter. They derive nutrient from dead and decaying matter. Okay? And bacteria and fungus are the main saprophytic organs. Bacteria as well as fungus are the main saprophytic organs. Bacteria and fungus are the main saprophytic organs. Some angiosperm also follow saprophytic mode of nutrition. Example, Neotia. Neotia is an angiospermic plants. They also follow saprophytic mode of nutrition. The roots of Neotia associated with mycorrhizae and absorbs nutrients as a saprophyte. So, the root of Neotia roots may associate with some mycorrhizal fungi and they absorb nutrients as saprophytic mode. Then Monotrapa, it is an Indian pipe. They grow on humus-rich soil found in thick forest. Monotrapa, they are found in humus-rich soil and it's absorb nutrient through mycorrhizal association. So these are the saprophytic mode of nutrition. Now parasitic mode of nutrition. Parasitic mode of nutrition. Parasitic mode means organism deriving their nutrition from other organism and causing disease to the host. So the organism, they derive nutrition from another organism and causing disease to the host. It is called parasites. So parasites are organisms, they derive their nutrition from other organisms but they cause disease to the host. There are two types, obligate parasite and partial parasite. Total parasite and partial parasite. Total parasite, also called as obligate parasite, they completely depend on host for their survival and they produce a structure called hostoria. Obligate parasites are of two types, total stem parasite and total root parasite. Total stem parasite and total root parasite. Total stem parasite and total root parasite. Total stem parasite means the leafless stem twine around the root and produce hostoria. The leafless stem twine around the host and it produces hostoria. These hostoria may absorb the minerals and water from the host and it may cause disease to the host. Example, Cuscuta. The total stem parasite example is Cuscuta. Then second one is total root parasite. They don't have stem axis and they grow in the root of host plants. So total root parasite is grow on the root of the host plants. Example, Rafflesia. Example, Rafflesia. Okay, these are about total parasite. Then partial parasite. Partial parasite is again divided into partial stem parasite and partial root parasite. Partial stem parasite and partial root parasite. Partial parasites are contain the plant of this group contain chlorophyll and synthesize carbohydrate. But the water and mineral requirements are depend on the host plant. So partial parasites are, they depend on the host for only water and minerals. They can prepare their own food and synthesize carbohydrates. The partial stem parasites, example, Loranthus. Loranthus are partial stem parasite. It is grow on the mango tree. We can found on mango tree and everything. They are green in color. They have chlorophyll and they can prepare their own carbohydrate. But they depend the host plant for water and minerals. Then the partial root parasite, example, Sandarum album. Sandarum album, that is sandalwood. So these partial root parasite in its juvenile stage produces hostoria which grows on root of many plants. So In juvenile stage, this Candida album may produces host area in the root and it absorbs water and minerals from the host plant. So these are about the parasitic mode of nutrition. Now we are moving to the symbiotic mode of nutrition. Symbiotic mode of nutrition. Symbiotic mode, we know that first one is lichen. Lichen is the symbiotic association between fungi and algae. Algae mainly includes blue-green algae or cyanobacteria. So it is a mutual association of algae and fungi. Algae prepare food and fungi absorb water and provide tannin structure. So algae green in color, they can prepare food. The fungi which absorb water. So it's a mutual association. The second one is mycorrhiza. Mycorrhiza. Fungi associated with root of higher plants, which also include gymnosperms. So mycorrhiza are some fungi, they associated with the root of higher plants. Example, pinus. Third one is rhizobium and legumes. We already discussed about rhizobium and legume. Rhizobium and legumes. This symbiotic association may fix atmospheric nitrogen. The last one is cyanobacteria and coralloid roots. The coralloid root of cycas containing a cyanobacteria called nosto and anabina. So it may help in atmospheric nitrogen fixation. So the association is found in cycas. It is a gymnosperm. Cycas, it contains cyanobacteria, nosto, some containing nosto and anabina also with the coralloid root. So these are about the symbiotic association or symbiotic mode of nutrition. Now we are moving to the last section, insectivorous mode of nutrition. Insectivorous mode of nutrition. Insectivorous mode of nutrition means the plants which are growing in nitrogen deficient areas develop insectivorous habit to resolve nitrogen deficiency. So nitrogen deficient area, the plants which is grown in nitrogen deficient area may produce the insectivorous behavior to resolve the nitrogen deficiency. First one, Nepenthes. Examples are, first one, Nepenthes plant. So Nepenthes. Nepenthes is also called as picture plant. Picture is a modified leaf and containing digestive enzyme. The rims of the picture is provided with nectar glands and act as a attractive lid. When the insect is trapped, proteolytic enzyme will digest the insect. Actually, this is a picture plant. This is the rim of the picture plant. The rim of the picture plant is highly attractive and the rim of the picture plant provide nectar glands. Here some nectar glands are there and the lid is highly attractive. So the insect may attract by seeing this, the insect may attract the plant by seeing this attractive lid and they take the nectars from this rim in the picture plant. So they are easily get trapped, and inside the picture plant there are several enzymes. Proteolytic enzymes are there. When the insect is trapped, the proteolytic enzyme will digest the insect. This is simply Nepenthes. Now Drosera. Drosera consists of long club-shaped leaves with tentacles that secrete sticky digestive fluid which looks like a sundew and attract insects. Actually, a club-shaped leaves is there. It contains several nectar glands. It is look like sundew. So this may attracted by the insect and may come to trap in this plant. So several enzymes are there. These enzymes may digest the insect and utilize the nitrogen from that insect. Then Utricularia. Utricularia is a angiospermic plant. It is found in aquatic nature. It is found in water. It contains some air bladder that collect the insect in the water. So Utricularia containing air bladder. So these air bladder may what it happens is, while water currents occur, in this water current there are several minute insects are there. These minute insects may trap inside this air bladder. This air bladder contains several proteolytic enzymes. These proteolytic enzymes may digest these small insects and they utilize the nitrogen and they resolve the nitrogen deficiency. Last one is Venous Flytrap or Diarrhea. Venous Flytrap or Diarrhea. Leaf of this plant is modified into a colorful trap. Two folds of lamina consist of sensitive trigger hairs, and which insect touches the hair, it will close and traps the insect. These are the nectar glands, and several hairs are there. Several minute hairs are there. While the insect come and touches this hair, or insect come and take this nectar, this leaf is automatic, or this leaf automatically close and which traps the insect. So the plant uses that nitrogen of that dead insect. So these are about the insectivorous mode of nutrition. So by this we are ending our chapter. If you have any doubt, you feel free to ask me. So that's all of mineral nutrition. Thank you, students.